Hello everybody, my name is John Candy. I am the section head of the Molecular Genetics Lab at DFO's Pacific Biological Station. The last time I talked to this group was in 2017 at the CEP Community Workshop in Canal. At that time, the Molecular Genetics Lab had just started on a large-scale application of new technologies for Chinook and coho stock identification. This is an update with the substantial progress that has been achieved to date. To start this presentation, I will explain some of the terms that we use in the lab. For stock identification, we use a number of different natural tags or genetic markers. Some of these markers have become obsolete as new markers are discovered. Allozymes was the first marker type for stock identification and predates the use of DNA. Here, variant forms of proteins can differ structurally but not functionally. So certain populations have specific variant of a protein, allowing us to tell one population from another. This marker type is not used by the lab with the advancement of DNA-related technology. Microsatellite markers use sections of the genome where there are repeat sequences of base pairs. The differences between populations are determined by the number of repeat sequences. Alternatively, SNPs, or single nucleotide polymorphisms, is another marker type where point mutations can give rise to alternative bases. This marker type has seen increased use in our lab. Genetic stock identification, or GSI, is a method of using one of a number of genetic markers to identify a fish back to stock of origin. This assignment requires a baseline of representative populations, usually over the range of the species. Parentage-based tagging matches individuals back to parents to determine hatchery and brood year. Because PBT is matching, this provides a high level of precision by identifying the individual parents. The language of DNA consists of a four-letter alphabet, A, G, C, and T base pairs. Here's an illustration of the difference between microsatellites and SNP markers. The microsatellite markers use repeat sequences of DNA. Here is a four-base pair repeat GATC, or what we call a tetranucleotide. Certain populations will be found to have more or less numbers of these repeat sequences. This can be used to identify one population from another. SNPs, on the other hand, vary at a single base pair, as seen in yellow. The frequency differences of many SNPs are indicative of a population. While microsatellites are from non-encoding regions of the genome, SNPs are abundant throughout the genome and can be from encoding and non-encoding regions. GSI, or genetic stock identification, compares individuals from an unknown sample, say from a fishery, to frequencies of known spawning ground samples representative of a population. Here we have three different colors representing three different populations in our baseline. A probability-based assignment of the unknown individuals to the baseline of known populations is based on the degree of genetic similarity. PBT, on the other hand, is matching unknown fish to parents. The two sets of parents have been genotyped, represented by different colored circles. Offspring have traits represented by different colors of circles, where one comes from the mom and the other from the dad. When one of the offspring is sampled at sea, parents can be identified by matching traits present in each of the mother and father. PBT requires genotyping all hatchery brood in order to successfully assign all hatchery individuals back to parents. The two methods, GSI and PBT, work together. First pass, we'll try to identify as many of the unknown fish matching to hatchery brood parents found in the PBT baseline. The remainder not matched are then identified by genetic stock identification to population and regional group from the GSI baseline. Now that you're familiar with the different marker types and methods used to identify salmon stocks, I'm going to present a timeline of changing technologies occurring in our lab over the years. Above the timeline are some key publications which changed the course of our stock assignment work. The first application of GSI occurred in the early 1970s using allozymes. This was first published in a paper in the mid-1970s. Our lab started using allozymes in the early 1980s, and the application was to identify Fraser from east coast of Vancouver Island chum stocks in the Strait of Georgia. This was to allow terminal fisheries along Vancouver Island without impacting Fraser chum. The use of allozymes died out with the discovery of PCR. PCR, or polymerase chain reaction, was discovered in the mid-1980s by an American biochemist who won the Nobel Prize in Chemistry for this discovery. PCR allows the replication of many copies of the same piece of DNA. So for microsatellite markers, PCR allowed us to amplify repeat sequences to determine size. As mentioned before, the number of repeat sequences are indicative of certain populations. In 1997-98, we analyzed our first mixed-stock Chinook fishery with 6 microsatellite markers. This was followed by the development of microsatellite markers for sockeye, coho, and steelhead. Microsatellites were the primary marker type used in the lab from about 2000 to 2015. They continue to be used in the lab, but at a significantly reduced as SNPs have become the preferred marker type. In 2006, 2 American fisheries scientists published a paper and coined the term parentage-based tagging as a method to identify potentially millions of hatchery fish back to their parental cross. As already mentioned, this requires genotyping all the hatchery brood. Given the current technology in the early 2000s, it was not thought to be feasible to genotype such large numbers of fish required to implement PBT. But by 2012, technology advanced further, allowing whole genome sequencing in a fraction of the time it took previously. We can now genotype by sequencing using SNP markers that allowed us to start in 2013 and 2014 PBTing for Chinook and coho. GSI using SNPs for chum started in 2020 and for sockeye in 2021. As mentioned before, technological changes have meant SNP markers are being used more than microsatellites, and here are some of the reasons for this. SNPs are highly automated, requiring less manual work for lab staff. They are well suited to high throughput, so we're able to genotype by sequencing large numbers of fish required for PBT. Greater automation allows repeatability between genetics labs and platforms. And greater GSI accuracy with SNPs allows us to assign individuals to finer genetic groupings. But the speed of turnaround is less for microsatellites between when the samples arrive at the lab and results sent off to managers. So microsatellites are still being used for in-season work, where managers often need results from samples the same day as they arrive in the lab. Next, I want to walk you through the steps in our high-throughput sequencing for PBT. Sample tissues for sequencing are collected and stored in Whatman sheets. At the top center of the slide, DNA is extracted and isolated from the tissue. PCR is used to amplify short, specific pieces of DNA about 200 base pairs in length. A cleanup step after amplification removes all the pre-products from the reaction, leaving the amplified DNA fragments called amplicons. These sequences contain our target SNPs. Next, barcodes are attached to the amplicon. Barcode is a manufactured unique sequence of base pairs used to identify individual fish once the amplicons are pooled for sequencing. Amplicons are loaded onto a semiconductor chip containing 160 million wells, with each well containing an amplicon. Once loaded on the sequencer, this semiconductor chip tracks the base pairs from each well as the sequencing proceeds, reading through the barcodes and to identify the fish and the target SNP. The ion torrent sequencer will genotype 500 unique SNPs for just over 1,500 fish at once. The preferred method of sampling DNA uses Whatman sheets to collect tissues. A small punch of tissue is taken from the operculum fin or adipose and stuck on this paper. Whatman paper is porous and allows tissues to stick firmly. Once stuck on the paper, drying the tissue will stop the DNA from deteriorating. For hatcheries, separate sheets are kept for male and female broodstock. Whatman sheets can be cut to various sizes for sampling at sea. Working under cover is preferred. This sampler has found an ice tray to be a handy way to move a tissue punch from the back deck to work inside. There are currently 61 hatchery broods that we have ongoing genotyping for PBT. Most of these brood sample collections are from major set facilities, blue color, but also from public involvement projects, orange, and community economic development projects, green. Many of these collections were started in 2013-14. Others have come on later, so depending on the age of return, we're now tracking second and third generation Chinook broods for many of the hatcheries. Annually, we're genotyping around 20,000 Chinook brood samples with a greater than 95% genotyping success rate. So far, we've covered the history of marker development and technological change in the lab. We've seen the move towards high-throughput sequencing of SNPs that allows us to genotype large numbers of brood and mixed stock fishery samples for PBT and GSI. Now I wanted to show you some of the results from the lab. This is an example of a northern troll fishery sample of coho catch from 2019. This is one way we report our results to managers as a table of percent assignments by regional groups arranged from geographic north to south. Baseline coverage for coho consists of 51 reporting regions where we have samples of known populations from southeast Alaska to California. In Canada, these reporting regions are conservation units for coho under Canada's wild salmon policy. In this example, a total of 298 fish were analyzed, of which 42 were identified by PBT, which means these fish were progeny of hatchery broods that we'd previously genotyped. The remaining fish are assigned using GSI, and the combined GSI and PBT estimates are reported by conservation unit. Typically in these northern troll samples, catches are from both Canada and US stocks. Highlighted in yellow, there is a considerable portion of the catch from mid-Skeena, Haida Gwaii east coast of Vancouver Island, and then US stocks Puget Sound, coastal US, and Columbia River. In another example, this one Chinook salmon catch, we are looking at just the PBT component. This is a 2020 Upper Johnson Strait Chinook sports catch. A total of 815 Chinook salmon were genotyped, of which 272 PBT identifications were made, or 33% of the sample. The most abundant proportion of the catch was from West Coast Vancouver Island's Robertson Creek Hatchery, consisting mostly 3 4-year-olds, a few 5-year-olds, and 1 6-year-old. Also a lot of Puntledge Falls 3 and 4-year-olds, Quinsam 3, 4, 5-year-olds, and Qualicum 3-year-olds, plus a small number of other hatchery stocks. Because we know the year the parents were spawned, we also know the age of each of these offspring at the time they were caught. The additional information provided by PBT and GSI feed into hatchery management by evaluating different survivals between hatcheries and between release strategies. It enables estimation of inheritable traits over multiple generations. It enables hatchery brood management for conservation and/or production goals, and it enables detection and monitoring of genetic and ecological interactions between hatchery and wild fish. PBT and GSI helps improve fisheries management by obtaining accurate measurements of harvest impacts on hatchery and wild populations. We can use it to limit harvest on endangered and threatened stocks in mixed stock fisheries, and it allows non-destructive sampling of sublegal catch. What can PBT tell us about broodstocks? Here are a couple of examples. By using all the 2018 and 2019 PBT assignments, straying between drainages where the straying Chinook are incorporated into the brood occurred at an average rate of 0.4%. Highest levels of straying occurred between adjacent drainages. Estimates of stray rates are important to determine the level of gene flow. High gene flow between populations can lead to an overall loss in genetic diversity. PBT has also been used to differentiate summer and fall run Punch River Chinook, helping to preserve the two separate run timing lineages. In 2018 and 2019, 4 to 5% of the fish previously selected as fall run brood were incorporated into the summer run brood, but only about 1% of the fish previously selected as spring run were incorporated into the fall run brood. Use of the PBT has been an effective way of identifying hatchery brood stocks for each of the run timing lineages. In summary, genetic methods provide a natural tag for determining stock of origin. Assigning unknown individuals requires a baseline and known individuals for both GSI and PBT. The baseline for GSI consists of representative samples from known populations, while the PBT baseline consists of all hatchery brood. PBT and GSI work together. First, PBT assigns individuals to parental crosses using the baseline of hatchery brood stocks. Then GSI assigns the remainder of the unknown fish to populations using the GSI baseline. Finally, PBT can tell us the parents, age, sex, and hatchery of origin of an individual from the parental crosses. I would like to acknowledge the following molecular genetics staff who provided all the innovative and exciting science to develop PBT and GSI for the use in Pacific salmon management. I'd also like to thank the many DFO, stock assessment, and CEP staff, along with Aboriginal groups, NGOs, volunteers who all provided the monitoring and sampling to make this work possible, and also the support of the Results Fund RF121. With science and CEP working together, we will better be able to inform harvest and production decisions. Thank you very much for listening to this presentation on recent developments in the molecular genetics lab in the use of genetic stock identification and parentage-based tagging.